What we're writing down right now. 1 minute. Like 1 more minute. Let's do that. Before we check our answers, I've been getting some emails about more study resources, which is great. You absolutely should be starting to think about studying. Our first exam is on October 4th, so that's a little under 2 weeks away. So good time to start looking over stuff. I know this class is really fast paced and not super conducive to doing all your studying the night before. Usually we find that people are like reading or studying or working on this course like 9 hours a week. The ones that do particularly well on the exam. Okay, so you should all have access to the SI worksheets. Does everybody know where those are? Okay, you can either get them in SI physically or they're posted on that module on Brightspace, and the answer keys are posted as well. Obviously try to do them first before looking at the answer key. Your book has questions at the back of each chapter. And I know that some people haven't found this mastering A&P study area, so I want to show you guys where that is, because that can be helpful as well. So those of you who have found it, hold tight. Let's see what my Brightspace looks like. So if you go to mastering A&P, in addition to the assignments that we've been doing, there are other resources. So there's this area called the study area. Has anyone seen this? No? Okay. Hopefully this is visible for all of you. I'm pretty sure it is. If it's not, let me know. But if you go to the study area, you launch it, you'll find that like broken down for each chapter, they have a bunch of different resources. So like, let's think cell metabolism. So they have tests, both a pre-test and a post-test. So like if we open up this pre-test, right, it has questions that you can go through to practice what you should be thinking about before the chapter. And then there's a post-test after. It also has quizzes of different types, right? So it has some multiple choice questions for you to practice on. It also has flashcards. I know people like to make Quizlets, but these flashcards for mastering A&P are helpful as well. Right, so if we look into cell metabolism, we create a deck of flashcards, and we won't need all these terms. Consult your outlines and what we've been talking about in lecture if you're not sure whether you need to be learning something or not, but you can go through them, right? It has these terms, defines them. So these can be helpful as you're getting started. So, I think they should be similar. I haven't looked through all the quizzes. I don't have a lot of experience using them to study myself because I teach the class, but I know that people who use this study area find it very helpful. So I think they are. Okay. So that's just an alert to know that that's there. Yeah, go ahead.
So the people that don't have books, you kind of get screwed.
For now.
It expires today or tomorrow.
Okay. I am in the process of talking to them. What I'm hoping they can do for you is maybe send you those access codes ahead of time. I don't know exactly how to get them to do that, but that's what I'm hoping to sort out this afternoon. And they still say they haven't shipped. Okay. Yeah, I've been calling people all week trying to yell at them, but nobody wants to take responsibility. Yeah, no, I got you.
I was just going to ask,
Yeah, they're multiple choice. I think we have them at 33 questions. You get the full time, so that leaves you a little under 2 minutes per question. There's an extra credit question at the end.
It's like it covers the beginning chapter, the one that we've done most recently, and then the next one, does it cover everything so far? Or is it just like chunk by chunk?
They're chunk by chunk just for the unit we're covering. You do have a final exam, which covers everything. It's super short. So what you should be focusing on is these unit exams. But yeah, you'll have to build on these concepts, right? Because we'll be talking about gradients in this unit. And then it's not like you can forget what a gradient is to understand nerves, but you won't be tested directly on the material again.
So do we have this in here that like combine this and the lab, or are they two separate ones?
Great question. So we have a final in here that is just for lecture material. You have a practical exam for anatomy. So it does make sense to think of it like a final, but basically that'll be tagged things on the cadavers and in the lab that has the bones and the models. So you'll be doing that separately, and there's no final exam for this lab. So it's just this one for the we also have office hours. Both the TAs and I have office hours. I'm happy to meet with you outside of these times as well. Just email me. I'm usually in my office and my door is usually open if I'm there, unless I'm blasting music, in which case shoot me an email or knock loud. Okay, and the final thing is the tutoring center. Just so you know where to find that, because I find that some people haven't used this before. Start tutoring here. Yeah, so you can schedule to meet online or in person using this. So if you go to the tutoring center website, you'll find a bunch of different information. So appointments, or we have drop-in tutoring. There's a schedule for that. So it's all color coded. The colors correspond to who is actually physically sitting in the tutoring center on that day. At least half of them are able to tutor you for A&P. So you just go and look in the subjects, right? So you can see Britt is doing A&P, Nicole is doing A&P, and so on. Other questions about that for now? We will also have a review day before the exam. It's not like you need to know this all tomorrow. I just want to make sure you have some resources to get yourself set. So before we go over the metabolism of fats and proteins, we'll go over that first question just about aerobic cellular respiration. Trying to remember where the whiteboard is. There we go. Okay, so we're going over the steps just to make sure that we remember them. So hopefully we had glycolysis as a step. Just weird on my screen. And then we had the linking step. Pyruvate. Maybe that was on your list or maybe not, but you want to remember that it does exist. So we'll put it here. And we have the Krebs cycle. And then we have our oxidative phosphorylation. You want to remember involves the electron transport chain. So what we really need to know for these are where we have ATP. So do we have ATP created in glycolysis? Thumbs up, thumbs down. Yeah, great. Another color. All right, so how many? Two. Yeah. Let's do this whole thing for 1 molecule of glucose. Okay, so where's the next place we see ATP? Do we see any in this linking step? Nope. Great. Krebs cycle? Okay, yeah. So we have 2 total here for each molecule of glucose. So if you look at a map of the Krebs cycle, you'll just see 1, but it has to go twice for each molecule of glucose. And then what about this last step, oxidative phosphorylation? Yes, we do. About how many do we have here? Yeah, about 30-ish. So how many of these steps, let's talk about the oxygen next. How many of these steps require oxygen? Just 1. And which one's that going to be? Yeah, this final step. Okay. This is where we require oxygen. So you might have said 2 because of the way that the electron transport chain backs up. So the Krebs cycle wouldn't run if there was no oxygen, but that's because it's backing up from this electron transport chain, not because it requires oxygen in and of itself.
Oh.
Yeah.
You said the Krebs cycle is twice for 1 glucose?
Yeah, and that's because we have the glucose splitting into 2 pyruvate molecules. All right. Pyruvate, pyruvate. So essentially we have like 2 linking steps, 2 Krebs cycles going on for each molecule of glucose. Right. So I don't want to guide you guys too wrong, but this is practically everything you need to know about aerobic cellular respiration. The only other thing that you really need to pay attention to is those electron carriers. So the NAD, the NADH, most of which is being created here in the Krebs cycle. Get a little bit of it everywhere else. And then it's all getting used in our electron transport chain, giving us NAD plus back, which we then use in these other parts. Something people sometimes get confused about because we talk about a lot of different types of metabolism, and we do talk about something other than just this aerobic cellular respiration flow, right? So what we did here was aerobic cellular respiration. Respiration is that lactic acid fermentation bit that we talked about as well. So that's not one of the steps here. That's if we're not able to do this, and therefore we're not able to do this, we wouldn't move the pyruvate into the mitochondria at all. We'd just loop back and try to keep doing glycolysis. So this little loop to just keep running glycolysis, that's the part with the lactate. Yeah.
So there was a question about how many ATP were created during the last step. Would 30-ish be an appropriate answer? If you want like the one?
Since it's multiple choice, just pick whatever is closest to 30. Yeah, basically. Yeah, because that is variable. So yeah. Okay. Questions? Any more questions about that aerobic cellular respiration? I know we talked about it like all in one day, which feels like a short time for cell restoration, but we do need to move on. Any questions? If as you're going through the material for this exam, you come up with questions, just make sure to make a note for yourself. Ask me. We can go over it one-on-one, or we can go over it on the review day. If you have a question, you're not probably the only person who has a question, right? Usually when I get a question, it means that lots of people are confused. So it's actually helpful for me if you tell me what your questions are, because then I know if there's something I glossed over kind of too quickly or something like that. So helpful for you to get the answer and helpful for me to get the feedback. Okay. So when we talk about energy, when we talk about metabolism, our main thing that we're focused on is the glucose, because that's our most common form of energy. That's our most immediate form of energy. And most importantly, your nerves, your brain can pretty much only run on glucose. But we are able to get energy out of other molecules than glucose. Now, we're not going to memorize the exact pathways for all the other molecules, but we are going to take a look at how this kind of interacts with our glucose metabolism. So I got a big map like this. Basically, what we're focusing on here, right, is that we can use both proteins and fats. So triglycerides here, or what type of macromolecule is a triglyceride of our 4 types? Is it a carbohydrate? Yes, no, no. What about a protein? No. Is it a lipid? Yeah, it's a lipid. Great. So triglycerides are lipids, they're a fat. So we can use those for energy as well. Basically, what we're doing when we're using these alternative forms of energy is we're taking them and pouring them somewhere in to our glucose metabolism pathway, right? So here we see our pathway that we've just gone over, right? Our glycolysis to pyruvate to acetyl-CoA to the Krebs cycle to oxidative phosphorylation. So this is all that we just went over. All right, so we can see as we break down proteins, we can kind of feed them into this process. We don't turn a protein into glucose, but we actually can turn some amino acids into pyruvate. We can kind of turn some into acetyl-CoA. We can feed others into the Krebs cycle. So we kind of skip earlier steps in this pathway, but then continue to use some of these later steps. The same thing is happening with our triglycerides, with our fats, right? Our fatty acids can feed into later parts of this pathway. Glycerol, the glycerol backbone of a triglyceride, actually, if you break it off on its own, is a carbohydrate. So that actually kind of feeds into the intermediate steps of glycolysis. So that's the sort of bird's eye view of how proteins and fats can be used for energy. We will look in a little more detail here. We're not going to look at any individual amino acids, but we're going to dig a little deeper on how this works. Before we get to that, we're going to talk about one of the ways that we can store glucose for later, because before we turn to alternative sources of energy like proteins and fats, your body is going to look to see if there's anywhere else it can find glucose. We do have a large polysaccharide, a big carbohydrate that your body uses to store glucose. All right, so we have a picture here of this molecule glycogen at the bottom. So this is a storage molecule. And what it's storing, right, is it's just linking together a bunch of glucose monomers, right? Monomer meaning one piece of something. So it's a polymer, a polysaccharide. It's just many glucose units put together. So if you have glycogen available in your body, this is one of the first places your body is going to go. We're going to tap the glycogen stores to start to release this glucose so that we can just do our normal aerobic cellular respiration thing if we're able. The places we store glycogen, we actually only have 2 places in the body that we're really going to do this. So the liver is the main source of glycogen. So that's the main place we'll see this glucose all stored up. The other place, the kidney. The kidney can do this as well. But if you think glycogen, really you mostly want to think about the liver. Now, there is kind of an intermediate molecule here that plays a role, something called glucose 6-phosphate. So when we're breaking down this glycogen, it doesn't immediately become exactly glucose. It's this kind of special modified form. Right, so it's this glucose 6-phosphate. And the reason we care about that is because glucose can leave a cell, but glucose 6-phosphate can't leave a cell. So in order to get glucose out to the rest of the body as we break down glycogen, we'll need an enzyme to turn that glucose 6-phosphate into normal old glucose that can use our normal glucose transporters. The only places we have that enzyme, which is glucose 6-phosphatase, right? ACE tells us it's the enzyme rather than the molecule. The only place we have those are the liver and the kidney, which is where we're storing our main glycogen stores. So if we have glycogen stored up in the liver or in the kidney, we can pull those glucose monomers apart. So give us glucose 6-phosphate. Then we can use our enzyme glucose 6-phosphatase to turn them into normal glucose so they can leave the liver. So in other places where we might feel a little bit of glycogen, we wouldn't be able to make that glucose leave the cell. We'd only be able to use it in the cell it's in. So that's why we really care about the liver and kidney and their glycogen stores, because that's the glycogen that we could liberate glucose from and then send it up to the brain, for example. The other neat thing, about glucose storage is that we're going to be able to create new glucose sometimes. And on a separate slide, we're going to break down a couple of really similar terms because we have a lot of words that start with glue here, and I don't want us to get confused, but gluconeogenesis is when we make new glucose. So gluco is the glucose part. Neo means new, and genesis is making something, right? So creating something. Now, if you see any terms that look kind of like this on the exam, I want you to slow down and really thoroughly read the word, maybe break it apart, because we're going to have gluconeogenesis, we're going to have, Glycogen Lysis, one more, I'm forgetting. Sure, I'll remember it in a second, but we're going to have a lot of words that look really similar, so don't just kind of glance at these long words that start with glue. So when we're making new glucose, we can make it out of glycerol, we can make it out of lactate, we can make it out of amino acids. And again, this is mainly happening in the liver, right? So we think about glycogen when we think about the liver, but we also think about this creation of new glucose in the liver. Sometimes we wave our hands and think about it as running glycolysis backwards, but it's a little trickier than that because there are some other enzymes involved. Mainly this glucose 6-phosphatase, right, to turn this glucose 6-phosphate that we may have liberated from glycogen into glucose. We can also go the other way as we're building up glycogen. We can add hexokinase to this glucose, giving us glucose 6-phosphate. We'll look at a map on the next slide, I think. Yeah, great. Okay. So our words. Right, so we had gluconeogenesis was what we had on the previous slide. I was making new glucose. And up here at the top of the slide, we also have glycogenesis. This is making glycogen. And then we have glycogenolysis, which is breaking down glycogen. So these are the three terms that I think you're most likely to mix up. So if you are going to make, actually, even if you are not planning to do any flashcards for this class, make these three. Just because those words are really easy to kind of trip over because they all look so similar. So if we go back and look at this map, right now that we have these terms down, right, what we're looking at here is our storage molecule glycogen. Right? So on the right-hand side, our glycogen lysis, lysis meaning breaking something down. So we are actually talking about breaking down glycogen here. When we break down glycogen, we follow this arrow. That gives us our glucose 6-phosphate. So if we want to stay in the cell that has the glycogen, we can turn our glucose 6-phosphate into pyruvate and just continue with cellular respiration. But if we want to ship glucose out to the other cells in the body, if we are, for example, in the liver, if we're in a cell that can do that, we don't turn it into pyruvate. Instead, we go into this little loop on the side. All right, so we can take our glucose 6-phosphate and turn that our glucose 6-phosphatase and turn that into glucose, which we could then send somewhere else, primarily the brain, right? That's usually where we're trying to send our new glucose. So this would be an example of that gluconeogenesis, right? Because we started with glycogen and now we're making new glucose from this storage molecule of glycogen. The other thing we can do, you're right, like eating a ton of pasta or something, right? Eating lots of carbs, maybe you can't use all that glucose right now. You need to store it as glycogen. So if we start with the glucose, right? So if we start here in the pathway, we'd use another enzyme, hexokinase, to turn the glucose into glucose 6-phosphate. And then we'd go up and turn it into glycogen. So this glycogenesis, right? Glycogen, unfortunately it overlaps, right? Genesis here. But that's the making of the glycogen. Questions about this linkage between glycogen and glucose here? We can touch base on this again later after you've had some time to mull over this. And we will be talking in the future about like hormones that tell your body when you should be making glycogen or when you should be making glucose. So we do kind of want to remember that these things can happen and that they usually involve the liver. But we will come back to this process. We'll hear its name again. The other, one of the other main places that we store energy is in fat. So most energy in your body is going to be stored as triglycerides, right? So for long-term energy, there's only a certain amount of glycogen you can hold. It's not that much, but you can store tons of fat. So we can break down those triglycerides to make energy, and they're actually really efficient. You can store a lot of energy in a triglyceride. They contain more energy per weight than carbs or proteins, so they are our most efficient way to store energy. So if we remember what a triglyceride looks like, right? It was a glycerol backbone All right, so here's my glycerol. And then we had our fatty acids. So how many fatty acids are here? Look at the word. Yeah, exactly. So we have our 3 fatty acids. So these triglycerides are going to be stored mainly in adipocytes. So those are just fat cells. All right, so we're going to go to the fat cells if we need to use this long-term energy. If we really don't have any glucose, we've used up our glycogen, we're going to go to those fat cells. So in order to process our fat and turn it into energy, we're going to have a couple steps here as well. So we're starting with a triglyceride here, right? So a fat. And the first thing we're going to do is we're going to rip that glycerol, that backbone, off from our fatty acids. All right, so rip off our fatty acids, right? And separate them. The enzyme that would do this is called a lipase. So ACE for enzyme, this like part telling you that that's how we're breaking down lipids. So that gives us glycerol and our 3 fatty acids. We're going to do different things with the glycerol than we do with the fatty acids. That's why we need to separate them first. So we'll follow the glycerol part first. Basically, we're going to send that glycerol into glycolysis and do our kind of normal remainder of cellular respiration. What we're actually turning it into is kind of an intermediate molecule. Don't worry about memorizing this molecule name, but it is dihydroxyacetone phosphate, but I don't expect you to remember that exact molecule. But what you do want to remember is that we're sending this glycerol intermediate into glycolysis. And so this is going to give us a fair amount of energy. Because it's a different molecule, it's not going to be the exact same count as for our normal glucose metabolism. So you'll see it's only making 20 ATP. Different molecules have different efficiency for producing ATP, and we don't need to know any more detail than that. But we get a fair amount of ATP here, right? So again, you don't have to memorize this number, just know it's like a bunch. Now the fatty acids are going to get broken down more than this. So that was our whole glycerol backbone was going into glycolysis. But then when we look at our fatty acids, we're going to do something called beta oxidation. So beta oxidation is basically our word for breaking down a fatty acid. And if we recall what that squiggle was, right, it's a bunch of carbons bound to each other. That was the points of our squiggle. And then they're bound to the hydrogens as well on either side to make like 4 of them. Do you guys have a question back there? Did I go too fast?
It's okay.
Okay. So here are hydrogens. Okay. So what we're doing with beta oxidation is we're taking this fatty acid, and we don't actually need the whole thing. We're going to take it in chunks of just 2 carbons at a time. All right, so this. And most fatty acids in our triglycerides in our body are like 12 to 18 carbons long. So it wouldn't be just 2 chunks. We keep going. Get, you know, like six or more sets of these 2 carbon chunks. So just that 2 carbon chunk is going to get turned into acetyl-CoA, some electron carriers. And that acetyl-CoA goes into the Krebs cycle. And so here we see our fatty acid going into the Krebs cycle. Right. So just with those 2 bonded carbons here, we're going to be able to do this whole end of cellular respiration, right? This whole end of the process. And this also gets us like a bunch of ATP, a fair amount. So again, not as much as you would get for a whole glucose molecule, but a lot more than you would get if you did just glycolysis, for example. Again, don't need to know the exact number, but this is why metabolizing fats can give you so much energy. This is why they're able to store so much energy in a really compacted space, is because every 2 carbons in that fatty acid chain is giving us like about half, maybe a little more than the energy we would get from a glucose molecule. Yeah, we do glucose first. Yeah, and we'll go over that a bit when we go to muscles, where we'll be talking about different different sources of energy that we can get to in the exact order, and some of it will depend on timing, some of it depends on hormones, but yeah, we prefer glucose. So we'll also liberate glucose from glycogen, but then we'll go to our fats and our proteins. Muscles will actually also start to break themselves down at a certain point. So just a note, because I know like the keto diet is kind of popular, at least sometimes I think it's still popular, right? This process does release ketones as well. So that's where these come from, this fat metabolism. That's just kind of a byproduct we get there. So our protein metabolism, we're taking our big protein, right? A protein is a polymer, right? It's all folded up, but it's made up of our 20 amino acids. Okay, so we're then going to pull out the amino acids by proteolysis. So proteo for our protein, lysis for breaking it down. We get our amino acids out of the protein, and then we continue to break those amino acids down. Depending on what amino acid you're breaking down, you'll get kind of a different resulting product. But pretty basically, we're going to get an ammonia group removed. Right? So ammonia will come off, and it'll then basically urinate it out. Your body will remove this waste product. And we're going to get this keto acid as the remainder of our amino acid. And that keto acid will either be converted into like pyruvate or acetyl-CoA, and it can go in around these steps of our normal metabolism pathway or cellular respiration pathway. This isn't quite as efficient as storing energy as a fat, because like amino acids are bigger. This is a little more difficult process. Your body doesn't prefer to break down like your skeletal muscle, but it can do it. So questions about either of these processes before we talk a little bit about cell membranes. Move our way into membrane transport. So we're going to skip this problem for right now, and we'll just go in directly into our membranes for time. But after class, try making sure that you can roughly draw a plasma membrane from memory, so that you can remember its constituent parts, right? We do want to remember the pieces of a plasma membrane, and hopefully you've seen them before. So when we talk about a plasma membrane, we talk about the fluid mosaic model. Mosaic because it's made up of a bunch of pieces, fluid because membranes are kind of jiggly, basically. So when we're talking, I suppose you guys can draw along with me, right? So when we're talking about our plasma membrane, the basis is going to be our phospholipid bilayer, right? So phospholipids, you remember, are a type of lipid, right? Remember, they have a specific property. What's special about a phospholipid? Exactly. So here we have our hydrophilic head. That's the phosphate group I've just drawn on. And then we have our nonpolar hydrophobic tails, which means they have to line up in a specific way, right? They have to form a bilayer. So by two layers, right? So they have to form this way because those tails are trying to get away from water. So they basically use those hydrophilic phosphate heads to protect themselves, to touch all the water in the external environment and in the cytoplasm inside the cell. So we're gonna have our bilayer. We are going to see another lipid involved in this. So cholesterol is a lipid. So cholesterol, it was the basis for like our steroid group, right? So we recognized it because it was kind of 4 rings linked together, right? So we had our cholesterol. We're also going to see super important for transporting things across our membranes are our membrane proteins. So membrane protein can stay on one side of the membrane or it can go all the way through. So I'm going to draw one that goes all the way through. Okay, so we have a membrane protein. And these are going to allow things to pass through the protein and therefore not have to touch these hydrophobic tails, which is how we're going to get charged molecules across, how we're going to get polar molecules across, because those are the things that wouldn't like to touch our hydrophobic fatty acid tails. We're also going to have some carbohydrates. So our carbohydrates are really going to be kind of tacked on to other molecules. So we might have a carb kind of tacked onto a lipid, or we might have a carb kind of tacked onto a protein. So those are our mosaic pieces as we start to build a membrane. Here's a better drawing. We'll go through their roles a little bit here. So the phospholipids are that base of the membrane, that barrier made up of 2 kind of opposing rows of phospholipids. Now, what their main role is to separate the outside of the cell from the inside of the cell. Now, that means they're going to block things that are dissolved in the water from moving between 2 of our body fluids, right? Our intracellular fluid and our extracellular fluid, right? Those are our parts of our total body water, which would separate it based on whether it's in a cell or intracellular fluid or ICF, or outside of the cell or extracellular fluid or ECF. So when we say water-soluble substances, we'll be thinking about this more as we talk about concentration gradients and electrochemical gradients and transport. But this is just a note here. So when we say water-soluble substances, 2 things that we're basically going to have that are water-soluble in our cells are ions, which are going to be charged. That's what we're going to be thinking about for the most of the rest of this unit, but also polar molecules. So glucose, right, is a polar molecule, and it's water soluble. It would not be able to travel across the membrane either. So anything with a plus or minus next to it, and our carbohydrates are going to be considered water soluble. They're not going to be able to pass easily across this membrane without help. Phospholipids do provide fluidity to the membrane. They can kind of jiggle next to each other. right? They're not covalently bound to each other. They're just kind of there spatially. So there's some movement possible in the membrane here. Okay. So because water is polar, this membrane is impermeable to water on its own if you have only phospholipids present in this membrane. Right, so water would not be able to slot itself in there and pass those hydrophobic fatty acid tails. But our cells are going to have these molecules called aquaporins. And I'll talk a bunch about aquaporins in the renal system in A&P2. So aquaporins are membrane proteins that let water get across. So when we talk about water moving across a cell membrane, which we will talk about when we talk about osmosis, we're just going to imply there that there are aquaporins allowing that to happen. So they sometimes seem like an exception to this rule. They're not actually an exception to this polar and ion kind of rule about not passing the phospholipid bilayer. It's just that we often just kind of assume that there's an aquaporin there because they're really widespread. They're pretty much everywhere. Our cholesterol is there. Humans are animals, so cholesterol is present in animal cells. We are animals. What it's doing is adding fluidity to the cell membrane. We kind of think of it as like a buffer. It prevents these membranes from either getting too jiggly, too fluid, or too stiff. This is because if we didn't have cholesterol, those phospholipids, while they're not covalently bound to each other, they might create a kind of crystal structure. They might find kind of optimal positions and try to stay there. And cholesterol just kind of breaks it up and stops us from doing that. They also do decrease the permeability of the membrane to water. Our integral membrane proteins, well, that's like super important thing, although I think we'll pick up tomorrow with the role of carbohydrates. The integral membrane proteins we'll be talking about a lot. They're going to cross our cell membrane here, and they're going to allow things to move through them usually. Because they have to go all the way through the cell membrane, just like our phospholipids, they're going to be amphipathic. So like if we looked at this part of our protein, we'd expect that to be hydrophilic, just like the phosphate heads that are next to it. If we looked at this part of the protein, it's touching those fatty acid tails, so we'd expect it to be nonpolar and hydrophobic. And then this part would be like polar again. And now you guys got places to be, so we'll pick up here, talk a little bit more on these proteins and our carbohydrates when we pick up on Friday.